So good morning or good evening, everyone. Welcome on our GWAS visualization course. So my name is Anastasia Sedlakova. I'm a senior community engagement scientist working in DNA Nexus focused on JupyterLab.
And I'm Ted Ladaris, so I'm a bioinformatics trainer at DNA Nexus.
And today we'll be reviewing the basic output file formats for GWAS. So we will be working with a Regini output, but we will be showing briefly how you can use the tooling. Also, we'll be exploring GWAS results in the LocusZoom app, and Ted will show you how you can plot and annotate interactive GWAS plots using Jupyter notebooks, like Python notebooks and R. Okay, so I just wanted to mention a couple of webinars that we already did. So why we decided to use Regini, even though we see that majority of you were using Plink for GWAS, is because we were showing the GWAS with Regini webinar, and this is kind of like a follow-up on that, how you can visualize your data when you do Regini analysis. And also in case you are just starting with JupyterLab and you don't know how to use JupyterLab, or you don't know what is Spark JupyterLab or single node JupyterLab, then we definitely advise you to look at our session, and you will find there a YouTube video and slides for that. So sorry for the animations a bit. So when we are looking at the full GWAS workflow, we see that we start with the GWAS analysis itself. So here you can see that we have genotype files and we have a phenotype files. So we are doing some QC, both on phenotypes and genotypes, and we are doing actually GWAS analysis. And the result of GWAS analysis will be at least when we have the p-values for each of the candidates, and we're trying to figure out what are the significant associations that we can work on with. So, and this is actually the focus of this session of this webinar, is that we're visualizing candidates. So most of you maybe know this is Manhattan plot. We will be looking at it in a few moments, and we'll be looking into LocusZoom App. And also what you can do is not only visualizing p-values and deciding which one are significant or not, but also annotating your results with different sources. So here we will be showing how you can use clean bar annotation for that. And also once you're done with that, you can also do polygenic risk score calculation and create a risk score. And as we saw in our poll in the beginning of the webinar, most of you are interested in us creating a session on polygenic risk score calculation. So stay tuned, and we will make a session specifically focused on polygenic risk score. So we will be showing here two formats, but as I said, we'll be focusing on Regini mostly. So here is how the Plink output format looks. So it's a tab-separated table where you have chromosome, position, ID of the variant, reference, and alternative allele. Then you have like what type of the test, if it's additive, and you have odds ratio. Then you have some statistics and p-values. And as noted here, the variables that are most important for us is some identification of the variant and also p-value. So we can have a decision process where we decide whether this association is significant or not. Regini output is a bit different. So let's look at the key differences. We will see in the later slides that the difference are in preprocessing for the LocusZoom, because LocusZoom was originally created to look for Plink format. So if you are using Plink, and we saw that majority of you do, then you can just freely start the LocusZoom app and you can use your output formats without any preprocessing. With Regini, it's a bit different, and we'll show you in a moment how you can do that. But the main columns will remain like similar, not the same. So you have a chromosome and position. You also have a p-value, although here it's a logarithmically transformed. You have a p-squared test, standard error, effect size. Also, you have a test you have ID alleles like reference. This is a reference allele, allele 0, and allele 1 is alternative allele. And here you see that there is a frequency for alternative allele. And this will be necessary for us to make QQ plots in LocusZoom. So I already mentioned LocusZoom several times. So what is it actually? It's web-based in JavaScript tool where you can see a rapid visualization. So this tool was not made by DNA Nexus, but DNA Nexus, let's say, amplified. So made a built-in app in UKB that you can use to visualize your results. So what it can do Manhattan plots, it can do QQ plots, some summary statistics. So for visualization, you need to specify input files. However, if you forgot some input files or you decided you want to look into different input files, you can just provide file ID, and I will show it to you in a moment. And here you will see an example usage. So this is a link to a tutorial. Here is a link to the LocusZoom app. So you will need to authenticate if you want to accept this link. And here is like the really very like very important thing that you need to do is that if you are used to use JupyterLab on GWAS has a default 4 hours time period. After that, it will just terminate. LocusZoom doesn't have this functionality. So each time you decide that you don't want to use your app, you need to terminate it. So just be careful so you don't end up with an app running for several days. So I will show to you on a slide just briefly how you can start analysis, and then I will do the short demo for the LocusZoom app. So how you can start analysis? You can go on a Start Analysis button, you select LocusZoom App, and the only thing that you need to, that you have to make an input is provide out files, which would be output files from Regini or for Flink. And also you can, for sure, on a stage settings, you can change the instance type. So LocusZoom normally runs with a normal priority, so not high priority, meaning that the app could be terminated, but most of the time, like you're not writing any code. So for me, I never run it on high priority because it saves money to run it on normal priority, which is a default setting. But in case you need to do that, for example, you are doing a demo or you have a presentation that you really need to have your app running, then I recommend you to run it on a high priority. So once your app is running, how you can access it? So you can access it, this is the print screen from the Monitor tab. When there is a worker URL and there is a URL address for that job, you can click on it and you can access your LocusZoom app. And here is a note. So even though the status will be running, immediately after there will be worker URL will be created and you click on it, it could happen then you will see 502 error that there is no service running there. So don't panic, just take a deep breath, make a coffee, make a stretch, and usually in a few moments your app will be running and you just need to reload your tab and you will see LocusZoom running. As I mentioned before, LocusZoom was made for Flink files. Here we are using Regini, but Regini files need to have some preprocessing. So what can we do with that? Here is the example script, and we put it into our repository. We will link to our repository in the I think that last slide or something like that. And when you will get the slide, you can go there and check these files. So we're assuming that you started your JupyterLab instance, you downloaded the whole directory containing output of the Regini GWAS analysis into one directory. And these are our assumptions. So how you can do that, like what you can do then with them for preprocessing. So Contrary with the link, regine has an output for each chromosome separately. So for blink, all the chromosomes are in one file. For regine, each chromosome is separately. So in order for you to look at the Manhattan plot as you're usually used to see it, meaning like different chromosomes together, you need to concatenate your regine files. So this is how we do that. We use the first line to create a header, and then we concatenate the rest. We need to add a pound key to the header. So let me just go back and show it to you here. So here, Plink have this pound key. And for LocusZoom, it means that it will look at this as the name of the columns. If you will not put the pound key as the sign, you will see just column 1, column 2, column 3, which is really inconvenient. So that's why you need to put a pound key to that. And also, you need to convert file from space to tab separated. So default for regine is space separated, and you need to move it to tab. So this is how you do it. So it's like 4 bash commands that you can use, and then your files are preprocessed. Here are the tips for preprocessing GWAS files for LocusZoom in case. So as I said, the header file needs to start with pound key. And also the limitation of LocusZoom on JupyterLab is that it handles files that are no more than 1 gigabyte. So what would you do if you have the file which is larger? So there are different things that you can do. For example, you can filter by excluding clients containing NA values. And this has happened to us when we were testing with the Blink files, that a lot of variants has NA values, and you can just filter them out. And by that, you can reduce your file. And also, LocusZoom accepts compressed files. So you can use gzip compression to that. And this is also will reduce your file. Or in case that none of it help, you can also separate files by chromosome. Okay, so as I said, now I will briefly show you how you can do that with in slides, and then we will do the same thing in a demo. So if you specified files before when you were launching the app, the files will be pre-populated here, as you can see. Or you can just use file ID you can just copy it, and I will show you how you can do it and where you can find it. So once you put it there, the file ID, or you selected the file, if you wait for a moment, the window will pop out, and you need to specify to map your columns to the columns here. So you specify chromosome, gene position, reference allele, alternative allele, and we also need to check in that the p-values are log transformed, because this is the case for regine. Then we need to specify some additional file options, like what is the effect size, standard error, and the effect allele is alternative, and we have a frequency for that. So here you can see the preview. Once you have accept options, you will see that it's pending from the pending status to ready to view. And here again, in case like it takes some time, just make a stretch, wait for a couple of minutes. Usually it would work instantly, but sometimes it could take some time, and it, like, obviously it will take time. Like the bigger is your file, the more time it will take. So once everything is working, you can see on your results page, and here you can see Manhattan plot for Plink for all of the chromosomes. We will be showing to you just 2 chromosomes for regine output, but what we want to point here is that you can even zoom in more using region page. So once you click on it, you can, for example, use her position and find your variants. And so dive deep into your results. So here you can also see that there is a annotation, and also you can see hits in a GWAS catalog. So you will have a link to the GWAS catalog here with more details about the hits. Also, what you will see is QQ plot. When you see here 4 different colors, it means that the p-value are are binned by the minor allele frequency. So just some thoughts about which p-values are statistically significant. So most of the time, what is suggested is controlling false positives by setting false discovery rate. And we have a link here which have a quite nice and easy explanation about what approach to use. And a last thing, which is also displayed apart from the summaries, like table for the significant variance, is a genomic control lambda, which basically shows you how your observed p-values differs from the expected p-values. And here you can see a link to the BioStars forum about more details with some code, how these values are exactly computed. And as we said, don't forget to terminate your LocusZoom because it could end up running forever and we don't want that. Okay, so now I will show you a demo for the LocusZoom. So everything is running. So this is what you see if you launch your LocusZoom. And here you have import, analyze, and about. So I'll start with the import. And I pre-populated, I selected one file. It's pre-populated here. But what if I would like to search something different? For example, this is a combined for 2 chromosomes, and I would like to use it only for 1. Moment, I can show it to you here, is that in my projects, here I have a GWAS results, and let's say that I want to do it for this file, which I didn't select it when I was launching the app, but I can copy file ID. You can see it here. So I copy it here. I paste it, and I know that the build is GRCH38, what I was using. So I pasted it here, and then I will map the columns. So just a moment. This will help. So so I'll show it to you as a pre-populated file like this. But for whatever reason, it doesn't work for me. So let's quickly look into monitor page to see what's happening here. So on the monitor page, I see that it's running. The job is running nicely. Let me just reload it. Select again, and I copy my file ID. Okay, so now it's working. So in case you have the problems like that, just reload it. And so this one is an unprocessed regime, so it will not work. As you see, everything is a column 1 because it's space separated. So what I will show you here is, instead of showing file ID, I will use the pre-populated file, and here you can see that the files are nicely. And just so you know how the files look here, so I'll go to manage so you can see the files. And GWAS results is what I have here. And so here we just look at the preview so you can see how it looks like. So these are my combined preprocessed files. Found key in the beginning, so LocusZoom knows that it's a header, it's top separated. Okay, so let's continue. So we have a chromosome here, position is gen position, ref allele is allele 0, and out allele is allele 1. And here we have a p-value, and it's log 10p, and it's log transformed. So here we have our preview. Let's click on the next. Although we can also select here, but we don't have to. It's okay for us to select, to just map these columns. This will be sufficient. We'll click on next, and here are the optional fields. We have an artifact size here, it's beta. Standard error is SE, And I forgot to mention, when we have on the slide where we have an example of Regini output, you will also see a link to Regini documentation when all of it is described what actually is in Regini output format. Effect LL is alternative. We have a frequency here, and frequency is specified by A1 freq. We have an accept options, and then we can submit. But I won't submit because I already did it. So I have a here. So here you see the analyze, and I already have this running. So let's look here. This is what I have here. I have a Manhattan plot, and let me just make it a little bit smaller. So here I have a Manhattan plot for 2 chromosomes, and I can look more into the region page. And here I can select some, I can dive more deeper, like selecting some coordinates. So I won't do that. So let me just reset it back to normal. And you can see. Here you can see that there is a threshold for significance value, which is 0.05 on 10 on 8. So it's like 5% significant values, and we have 1 million in SNP. This is how the threshold was constructed, and we do not have loci above it. So that's why top loci table is empty here. But we have a QQ plot here, and you see that the LLs are binned based on the minor LL frequency in 4 categories. And here is a confidence interval, like not line, but the confidence interval of something which is like in line, observed, and expected. And here you have a genomic control lambda, where we were talking about the coefficient that explains to you difference between observed and expected values. So this was the demo for LocusZoom, and now Ted can continue with showing you really very cool R notebooks.
So let's kind of talk about bringing our results file into JupyterLab and working with it in R. So I'll give you a quick tour before we actually get into the notebook, just to orient everyone to how we're going to go about it. So when we're working with visualizing GWAS data with R, we're going to be using DX JupyterLab. And as Anastasia mentioned before, there is a seminar on that if you want to learn more about how to start it up and everything. So we are going to be using a couple of very helpful visualization packages that directly take these kinds of tables. So the first is this QQman package. And then the second is this Manhattanly Plotly package. So the main difference between this is that Manhattanly is built on top of a package called Plotly, which lets you kind of add more kind of interactive elements such as tooltips. So I'm going to be demonstrating the R notebook. We may go through the Python notebook if we do have enough time at the end, but we do want to have time for questions. So both of these are available on our UKB Wrap GitHub repository, and these are links to this. And remember, we'll have the slides available for you, and there'll be a post on community as well about this. So these are kind of the two ways of like basically generating Manhattan plots. And so this is the Manhattan function from that QQman package. And you can see that basically much like LocusZoom, you need to map in the column IDs into this function. And we'll get into this more in detail. But as you can see, this generates that Manhattan plot that you might be used to. So, and the key difference, again, is with Manhattanly is that you can actually add in custom annotations when you are exploring the data. So when you molecular points that might be of interest, like for example, you can annotate whether it's a clinically significant variant or not from and we'll be doing an example of merging our GWAS file with ClinVar. And also, the same kind of analyses that you do in LocusZoom is available to you here. So this is basically a GWAS QQ plot of the GWAS p-values again. And this is kind of the interactive version of that. Finally, and this is where we're going to be heading at the end, is that we're going to be merging our top candidates with ClinVar annotation. And so you can see that we are going to be displaying with the tooltip both whether the clinical significance of that particular variant, whether it was how it was called in ClinVar, and like, for example, the beta value as well. So this can be customized, as you will see. So I'm going to hop into the JupyterLab instance, and I just want to make sure that everything is running, that I haven't been signed out. So what I'm just going to click on this just to make sure that everything is great. So here we are in the JupyterLab notebook. So again, these notebooks are going to be available to you when we get on GitHub, and the link is going to be available. So let's kind of talk about where we're going. So we have a merged Regini file here. So what we need to do is we need to basically download it into the local JupyterLab worker. So if we look in this GWAS results folder, you can see we have some different output files. This is the one that we are basically going to download, and we're going to be doing a little modification to it so we can load it into R. So this right here, this is a little bit of bash code that basically is going to download our file from our project storage into our local storage. And then we're going to be looking at the first few rows of the file. And you can see that we've made, basically this file has been formatted for LocusZoom. So we need to, and you can tell that because there is this pound sign in front of it, in front of chromosome here. So we actually need to remove that for the for loading it into R, because R does not like these kinds of characters, special characters. So we're going to be using a sed statement to basically remove that pound sign. And now we're ready to kind of load things up. So these are the sets, these are the packages, and this is an install statement. So just want to note that these are not installed in the Jupyter instance by default. This is because of licensing issues. And I know that, there are some pharmaceutical companies that are on this call or webinar, and just want to make sure that you are aware of the licenses that are attached to these packages. So I have already installed these, as you can see. So what I'm going to be doing is loading up I've loaded up the packages with this code cell. And now everything is loaded. So now we're going to read our table into memory. And so we are going to basically use read.table on our file, which is in the local storage here. And basically we need to specify that the header is true because there is a header. We'll use this as is argument. And then we need to specify the delimiter using the sep argument. So if we do this and we just run ahead on the data frame that we've loaded, you'll see that we have loaded it correctly. And note that there is no # in front of chromosome here. That's because we removed it with that sed statement. So now we've got it in. Let's do a little bit of filtering and data transformation. So here you can see that this is actually the -log10 p-value. So we need to transform it back into regular p-values. So that is what this step is doing here. So we're taking our original data frame, and then we are going to basically add a new column that transforms it back from -log10 p-value. So if we run that, you can see that it is very similar to the previous data frame, but we've added this p-value column here. And there was a question about why our p-values are so high. And just keep in mind that this is really for demonstration purposes. Don't use, don't take anything here this was kind of done just really for demonstration purposes. So don't use these p-values for discovery. So one thing you might notice here is that there is this test column. And one thing about Regini is it tests multiple kinds of models. So we want to actually basically use only those rows that have that are additive tests. And then we will drop any NAs just to make sure that we're only plotting p-values that are not NA. So I'm going to run that. And if you just take the dimensions of that data frame, you'll see that we've got about 27,000 rows. And if we look at the first few rows of our data frame, you can see that, it's hard to tell here, but we did do the filtering here. So as was noted, the p-values here are relatively large. And this is kind of the lowest p-value in this set of variants. So again, please just take this with a grain of salt. So now we've got our data frame loaded. We've added that extra p-value column, and we've filtered out to basically the additive tests and removed any rows that had NA values. We can generate a QQ plot. And we can do this with that QQ function here. And we can just pass in that p-value column. So again, this is just a kind of normal QQ plot that you would generate with R. But let's talk about kind of the, so now we've kind of seen our QQ plot. Now we want to plot a Manhattan plot. So this code block is just basically setting some color palettes so we can distinguish the different chromosomes in our plots. So these are just basically a set of color values. And then we're going to basically be setting, we want our plot to be wide and high, so it's easier to kind of visualize. So this is that function that I was talking about, the Manhattan function, and this comes from the QQman package. So we're going to pass in our data frame. And remember, we have to actually map our columns. So for the CHR argument, we need to tell it that the Chrome column in our results file corresponds to that. Same thing with this base pair argument here. And we need to pass in the SNP ID as well, and then specify our p-values. So if we generate that, I just pre-generated this just to save us a little time here. And you can see that we basically have, our results file only contains 2 chromosomes, but you can see that there is chromosome 1 here and then chromosome 2. and they have slightly different coloring, so you can basically tell which p-values belong to which chromosome. So what if you wanted to just show chromosome 1? So we can do that by filtering on our GWAS additive data frame. So we can basically filter our data frame. So we want to select all of those. So we only want to select chromosome 1. So we can use a filter operation here to select the those rows that only have chromosome value of 1. And this is just to note that this is a character or string, because remember that you know, the autosomal chromosomes do range, are numerical, but there are also X and Y. So just keep that in mind. So we're going to filter that, and then we can pass this new data frame, this GWAS additive underscore 1, 2, and then we can pass that into our Manhattan plot. So if we run that again, our Manhattan function, you can see we only have the chromosome 1. Okay, so these are like kind of static p-value plots, but you know, R is kind of capable of a lot more. So I want to show you how to basically generate interactive plots with Manhattanly. So here again, we're going to basically produce a subset of our files, and then we need to, instead of Manhattan, the function is called Manhattanly, and the arguments are very similar. But the cool thing about this is that you can actually pass in annotation columns, and we'll utilize this in just a little bit. So I'm going to run this. It will take a second. Okay, so while we're waiting for this to generate, I'm just looking at Q&A. So can scripts be modified offline prior to starting an analysis? So the short answer, Kiara, is you can modify scripts, you know, using your own local JupyterLab instance, for example, and then you can upload it onto RAP if that's what you want. You know, I guess that can, you know, when you're kind of putting something together and testing it, it's oftentimes a little more comfortable to kind of test it locally. So yes, you can basically, you know, take a notebook that you've kind of worked with. Oh, and so that's just, hopefully that answers your question. Okay, so we generated our Manhattan plot. So now if we mouse over these individual p-values, you'll see that basically we get some information about that particular variant. So you can see that there is an ID column, and we're also displaying the chi-squared value and the beta value here. But again, that's the cool thing about Manhattanly, is that you can pass in custom annotations here. And we're going to be taking that advantage of that in just a second. So if you want to generate an interactive QQ plot, you can use a similar function in the Manhattanly package called QQly. I won't show that because it's going to take a little time to generate. So here, we're going to first subset our results, and we're going to set a threshold. And I know for those who do, you know, GWAS analysis, this is a very high threshold. But again, this is just for demonstration purposes. So I'm just going to run the cell. And we're going to arrange by basically by lowest p-value. So here you can see that with the dimensions here, we are left with, we started out with about 27,000 variants. Now with that threshold, we have about 71 variants. And so you can see this is the resulting filtered file. So we need to basically add columns to this data frame. So we're going to basically take annotation from ClinVar and basically merge it with this file here. So this is a little bit of code that basically will get kind of the latest variant summary from ClinVar here. So I'm going to just run that and it will unzip it so we can utilize it. So that's going to take a little bit. And thanks for all of your questions, everyone. You know, it's always really helpful to see what problems people are or questions people have. And again, we use that to really kind of inform things. So you can see that in our local storage, we now have that variant summary file. So that is a tab delimited file. So we need to read it into R as a data frame. And that will take a second. I think it's about a 93 meg file. So yeah. And then we can check out the call names. Just to give you an idea of where we're going. So we're going to only filter to those that are SNPs here. And then we are going to basically cast, make sure that our chromosome column is a character, so we can merge them. And this is taking longer than I thought. I should have preloaded this, so apologies. This should take a little bit of, it takes a little bit of time, but okay, so now it's ready. So we're going to be doing that filtering step that we just talked about. And so here we can take those top candidates. So those 71 candidates that we had in that above data frame, just making sure that our Chrome column is a character. And then we can use this inner join function. So this is a function within dplyr. And so we can basically, we're going to merge our top data frame, our top candidates with the ClinVar data frame. And then we need to give it a mapping. So on the left side here, you can see this is like the name of the column in the what's called the left table, which is that original file or original data frame, that GWAS top data frame. This is the name of the column in the ClinVar data frame. Here you can see that we not only merge on chromosome, but we merge on gene position. So that's what we're doing here. And then we can back transform our Chrome column into numeric here. And I can't remember why I did that after I gave the spiel. So let's just do that merge. And so you can see that basically these are everything up to this p-value. So these are just the column names here. And you can see that basically we've glued on all of the different columns from ClinVar. So if we do just a head, so you can see that what we have done. Unfortunately, with the data frame abbreviation, we don't see all of the columns, but you can see everything on the left came from our Regini file, and everything on the right came from ClinVar. So we've merged our ClinVar and our top candidates into this data frame called GWAS top annotated. So we map the same arguments that we did before, but now we can actually pass in an annotate for that annotation one. We can pass in clinical significance because it is now a column in our data frame that we're passing in. So this should take a lot less time to generate, hopefully. So here, basically, you can see that there are a lot fewer candidates in our Manhattan plot here, but you can see as we mouse over that we have that clinical significance in our tooltip. So again, this is kind of a nice way to kind of work with your collaborators to kind of find out more information about an interesting looking variant in a Manhattan plot. So if we wanted to save this and get it back into our project storage. So what we can do is we can basically take that data frame and use write.csv to save it locally. And then you can see that we do a DX upload to get that back into our project. So we are going to put it into our GWAS results folder. So if I run that, it's going to take a second. And if we look in our GWAS results, you can see that we have that ClinVar annotated candidates. So that was it for the demo. There were a lot of questions. So Ali Reza is asking a question. Could you introduce the best way package to find gene for each variant? So Ali Reza, what I will say as a quick way to do that, if you look in the ClinVar column, if you merge in with ClinVar, you can see that there is basically a gene ID and gene symbol column there. So that will map if the variant is within a particular boundary. That's done by ClinVar. So that was it. So we basically took you through kind of doing some interactive exploration with LocusZoom, and then we showed you the JupyterLab way to kind of load the results files into R. Again, I want to note that there is an equivalent Python notebook. It does not have the interactive plotting, unfortunately, but these are just kind of static plots. So this is also available to you if you prefer to work in Python. So just a quick note about our community. So definitely encourage you to join community. And as Brenton said, if you have sat in on this webinar and you want to join and do work on UKB research analysis platform, we've kind of given you the resources to do so. Keep an eye out on community forum. So when we send out, you will get a link to slides and everything, but any kind of updates we'll post on community forum. If you want more information but via newsletter, you can do that. And then we have 2 GitHub repositories here. So one with our example notebooks, and then some with material from the webinars.
Yeah, and sorry.
Go ahead, Amasaj.
I just wanted to go through the questions, but we can go do it afterwards.
Okay. Yeah. So that's it. We don't need to go through learning objectives again. Okay. So why don't we move forward to Q&A?
Yeah. So I just want to point from the answered questions that we have. There was a question about, can one also annotate both LMM output? And yes. So as our colleague Andre answered, is that everything about the file format. So your file should be tab separated. There should be bound key in the beginning. So, and you should have p-value, either log transformed or not transformed p-value. chromosome and position, some identifier of your variant to figure out variant annotation and to annotate it. So yes, and there was also a question about, for example, if you can use different colors, is that I think that most of the functions that Ted was showing has a parameter call where you can submit, and you were showing it in one of the code examples, where you can submit vector of colors and you can modify colors for your liking.
Yeah, let me just highlight that. So this little bit of code here, so this is where you can specify a custom color palette, and then you can pass it into that call argument for the Manhattan function.